Hello, good morning, good afternoon, good evening, depending on where you're joining us from in the world today. I'm Andrew Harris, I'm Director of Campaigns and Alumni Relations, and I'd like to welcome you to our latest Bright Minds webinar series, brought to you from Sheffield. If you've attended Bright Minds before, you'll be familiar with the series and why it was created. For those of you who haven't joined us before, the purpose of these webinars is to showcase the varied, the exciting, the truly impactful research taking place at your university, and to hear from current researchers at Sheffield who want to share their work with alumni and friends. Our webinar today, I feel, is a very special edition, as our work in tackling neurodegenerative diseases is one of the university's stated flagship research areas. It's also an area where we have global recognition, have seen some really exciting developments take place, and much of my team's fundraising work is geared around helping to accelerate the difference that we can make as a university to conditions like motor neurone disease, MND, or I believe ALS as it's called in the United States. I'm delighted to introduce today's speakers. So our first speaker, Professor Dame Pam Shaw. Pam is an academic neurologist and a world-leading researcher and clinician in motor neurone disease. Dame Pam holds and has held extensive leadership roles at local, national and international levels, including being the director of the Sheffield Institute for Translational Neuroscience, so CITRAN, since 2010. And Pam was a member of the university's executive management team as faculty vice president. Our second speaker, and the focus of today's webinar, Dr. Tobias Moll, and I think Toby's PhD was funded by philanthropy. Toby's a postdoctoral researcher in CITRAN, and his talk today is entitled Piecing Together the MND Puzzle, from Genetic Discoveries to New Treatments. Toby will discuss how genetic discoveries have helped us identify new methods for combating MND, and more specifically about his own research into 3 MND genes and how the results of this research could help to improve the treatment for patients with the disease. So today we'll begin with an introduction by Dame Pam, followed by the presentation from Toby. And after the talks, you'll have an opportunity to put your questions to Toby and Pam, and we expect this to last for about an hour. Before we get started, there are a few housekeeping items to allow you to participate to the full in today's event. So for you to have the best viewing experience, let's have a quick look. Forgive me, there we go. For you to have the best viewing experience, we have turned off microphones, and also we have enabled the chat function. So, if you do have any questions, throughout, please feel free to use that chat function. So it is the chat function we will use today, and not the Q&A. There have been questions submitted in advance, and Pam and Toby will address as many of those as possible. Please stay at the end of the talk because, there will be an opportunity for you to hear a little bit about how you can get involved more in this work. So enough from me. If I may, I'll hand directly to Dame Pam. Dame Pam.
Thank you, Andrew. I'll just share my screen. So good day. I'll say good day because our alumni are coming from many parts of the world. So a very warm welcome to this Bright Minds webinar. So I'm Pam Shaw. I'm a clinician scientist in neurology. And before Toby tells you about his work, I'm just going to set the scene for MND research, which is one of the big neurodegenerative disease areas within Citran that Andrew has described. So Citran was opened by Her Majesty the Queen in 2010. And really it was MND patients that brought forward the concept for this and raised a lot of money through philanthropy to make this beautiful research institute happen. And what we try to do within Citran is harness all the exciting developments in neuroscience and then translate these scientific insights into benefits for patients with neurological conditions across Glossop Road in the Hallamshire Hospital in the clinic. And we were really delighted that 10 years after the inauguration of Citran, we were awarded the Queen's Anniversary Prize for our impactful research for patients suffering from neurodegenerative conditions. So these are the main diseases that we work on within Citran, the three big neurodegenerative diseases, so Alzheimer's disease, Parkinson's disease, and MND, but we also have teams that work on stroke, cerebrovascular disease, and neuroinflammation, particularly multiple sclerosis. And we're driving forward research for patient benefit in all of those disease areas. It was a dream for me to be able to design our research institute, Citran, and we designed it so that we would be able to deliver benefits for patients. So we have multidisciplinary teams of scientists, but clinicians under the same roof, and the scientists and clinicians talking to each other. And these different teams really make sure that we don't get stuck when we're trying to unravel the disease mechanisms that happen. So when we discover a gene, we need to do the downstream biochemistry and so on to understand what that mutant gene does. And all of our science feeds into our drug discovery suite and our gene therapy suite. So we really are trying to harness scientific insights to generate better ways of treating these disorders. I got interested in MND as a young neurologist because I just thought it was the worst disease in medicine. It always seems to happen to the nicest of people who are often very active. So this is one particularly poignant example from Yorkshire. Rugby player who played...
Where it's grinding it. The Olympics game that could take a 59 minute before we get a winner. They really should have pushed up. It's going to be a good concentration defense. I'm afraid I'm not getting the pro forward to the
Just look at that.
They're aware of this run.
So magnificent little halfback. You can see the quality of Rob's motor neurons as he was scoring that fantastic try. This is Rob in 2023. He's developed motor neuron disease. His teammate and friend, Kevin Sinfield, carried him across the finish line of the Leeds Marathon. So he's now like a rag doll, really almost paralyzed with this very cruel disease. So I won't go into great detail about this, but we designed Citran to make a difference, and I think we really have. We've attracted a lot of funding. We continue to have a really loyal group of patrons who support research equipment and fellowships and PhD studentships through philanthropy, which really helps us. We run a lot of clinical trials for patients. We're training a lot of masters and PhD students, and we're publishing in the very best medical and scientific journals. So without I think the success is recognized nationally. We've had the Queen's Anniversary Award that I told you about. We won the NHS Future Parliamentary Award for our groundbreaking work on multiple sclerosis. We've had the British Neuroscience Association Outstanding Contribution Award for Neuroscience just very recently, and we've been invited by the BBC to describe the work of Citran on Jim El-Khalili's Life Scientific program. So I think it's doing well. And I'll just show you some brief examples of the human impact of what we do. So we're trying to harness that science to generate positive outcomes for patients with MND. And we're trying new neuroprotective treatments, but also better ways of managing the troublesome symptoms. So in MND, patients usually die from weakness of their respiratory, their breathing muscles. And we did a trial of this non-invasive way of supporting the breathing muscles with a mask system, a bit like people with sleep apnea would use, that they can put on when they go to bed at night. And this really makes a huge difference to survival, life expectancy, but also quality of life. And that resulted in a nice guideline, which has really changed clinical practice, not only nationally, but internationally. Patients with MND often get weak neck muscles, and if you put your own chin down on your chest for a few seconds, you can see how uncomfortable that is. Traditional collars don't work very well. So with patients, with bioengineers, material scientists, even fashion designers, we developed this head-up collar that patients tested and gave us advice on, and that's now available throughout the NHS and commercially available worldwide, the head-up collar. And this, one of the first patients who helped us test it, Philip, said he couldn't imagine life without it. We've been developing in our drug screening lab, developing new drugs, small molecule drugs, and this particular drug, M102, is targeting a stress response pathway that isn't working properly in patients with MND. And we've spent several years taking it through all the steps to get it into human trials. So it has orphan drug designation by the FDA and the European Medicines Agency. We've tested it in our preclinical models within Citran. We've partnered with a US biotech company called Eclipse Therapeutics and raised £5 million with them to do all the safety and toxicology testing needed to get it into human trials. And it's now poised to go into human trials. So within Citran, if you think at the top of this slide, if you think of the drug discovery pathway from identifying the target through all the phases of clinical trials to a licensed therapy, what we try to have is a pipeline of therapies in development at different stages. And we're very lucky that Citran acts as a magnet, really. So multiple biotech and pharmaceutical companies join forces with us to get these therapies through into the clinic. So we keep an eye on that pipeline all the time. We're very delighted with our advanced therapies, and many of our alumni will have heard of the Gene Therapy Innovation and Manufacturing Center, GTIMC, that was a big infrastructure development recently within the university. But one of the advanced therapies we're most proud of is tofersen for a particular subtype of motor neuron disease that have a change in the SOD1 gene. And we did all the preclinical work to show that lowering the level of SOD1 might be therapeutically beneficial. And then we were the UK site for taking that therapy, that approach, into human studies. And the different phases of the tofersen trials were published in the New England Journal of Medicine. And with all the phases of clinical trials, tofersen works. It lowers the level of the SOD1 protein. It lowers particular biomarkers, and it's beneficial to patients. So I've done more than 25 clinical trials for MND patients. This is the first one where patients are saying, I'm getting stronger with this therapy. It was licensed as a therapy by the FDA in the US last April, and just in the last 2 days, the European Medicines Agency has also supported it. And I'll just show you Les, who was the first patient that went into the tofersen trial in the UK. When he first started, he was in a wheelchair. He could just about bear weight on his legs with 2 elbow crutches, but really couldn't walk any distance. So when he was in the trial, what he found was he could go up his garden steps after a few months in the trial, still needing some support and help. But he hadn't been able to do that for 2 years. I'm so proud of himself when he got to the top. You can see it really effortful for him though. And then at the end of the experimental period of the trial, which he'll show, walking around his house, looking pretty normal with his gait without any walking aids at all. So he was one of the patients that really noticed that his muscles were getting stronger. And he's been having monthly lumbar punctures to deliver the tofersen treatment for more than 7 years now. So that has had our clinical teams very, very excited. And just to end, really, one of the things we take particular pride on in Citran is bringing on the next generation of researchers from both clinical and science backgrounds. These are the 59 PhD students I've supervised, and of course, Toby was one of the best of them that you'll hear from in a minute. So we do take, career development of our young researchers very seriously. So we have clinical neurology trainees that have got various fellowships, young neuroscientists that have got various fellowships, including the prestigious UKRI Future Leader Fellowships. And of course, Toby has just very recently had success with an MND Association fellowship to develop his own research programs. But we steer people and nurture them through all the career stages through to professorships, and we're very proud of doing that. Citran's success, we built it to double our capacity. We've actually more than tripled it, so we're absolutely full to bursting point. Fortunately, there is some university land next to Citran. We can't build the building higher because of the residential properties behind us, but we can put an L-shaped sister building alongside, and we're embroiled with raising the funds for that now. The Vice-Chancellor has charged us with raising £10 million through philanthropy, and then the university will support the rest of the funding required. And working with Andrew's team, we're working hard to generate that philanthropic income. So we're full to overflowing. We don't want to get stopped in our momentum in terms of delivering better treatments for patients. We want to continue to attract great people from around the world. We want to continue to raise the profile of Sheffield University for neuroscience research, and we don't want to have to stop these collaborations with pharmaceutical partners, because that's a very good way of getting treatments to patients. And we want science is rapidly moving, so we want to continue to have cutting-edge scientific facilities. So in the interests of time, I won't go through all of this, but I think we've got a very exciting and impactful future for neuroscience at the University of Sheffield. And we've already made some difference for patients, and we'll continue step by step to do that if we can, especially if we can invest in our new facility. So we've still got we've done well, but we've still got about 3.8 million of philanthropic funding to be raised. If any of our alumni can help in any way through contacts with people who know about these diseases through charitable foundations, we would be immensely grateful for your help. So thank you so much for attending our Bright Minds webinar, for your interest and for giving your time to be present. And we're very excited to have your interest and support. So thank you very much for listening. I'll stop sharing my screen.
Okay, should I go ahead and start the presentation?
Over to you, Toby.
Thank you. Okay. Can everyone see my screen okay?
Absolutely fine.
Great. Okay, so hi, so my name is Tobias and I'm a postdoctoral research associate at the Sheffield Institute for Translational Neuroscience. Now, for almost 7 years, my research has been focused on motor neuron disease. And in my talk today, I will discuss what the discovery of new gene mutations has taught us about MND and how this might lead to treatment strategies in the not so distant future. Okay, so just a brief introduction into my background. So I began my PhD in Professor Dame Pamela Shaw's group in 2017, and this was under the day-to-day supervision of Dr. Jonathan Cooper-Knock. I completed my PhD in 2020, so this was at the height of the COVID pandemic, and then I was fortunate enough to be awarded a postdoctoral fellowship, which was funded by a very generous patron affiliated with the university through his company called Kingsland. I then went on to begin another postdoctoral scholarship. This was funded by the US-based foundation called Target ALS, which I'm still working on. However, I'm due to begin my MND Association Fellowship in April of this year. So this is the transition point in my career, I like to think of. So it's my first shot at like an independent research track. So as I'm sure you can imagine, a career in academic research can be quite time consuming. However, when I'm not in the lab, I've particularly been enjoying my running in the past year or two in particular. I was born into quite an artistic family, so I grew up playing piano and violin, which I still enjoy today, and I studied ballet for 18 years. So as Pam's obviously touched upon, so motor neuron disease is a relentlessly progressive neurodegenerative disorder, and it affects approximately 1 to 2 per 100,000 people worldwide. At any given time, there are approximately 5,000 people in the UK living with MND. Now, although this might seem quite rare, estimates have the risk of developing MND as high as 1 in 300. So there is currently no cure, and patients who succumb to the disease tend to survive approximately, on average, 2 to 5 years from the onset of the first symptom. Now, despite there being no cure, we do have sort of FDA-approved drugs at our disposal. So the first being riluzole. So this extends survival by approximately 3 months, and more recently, edaravone, which slows down the progression of the disease in selected patients. However, I will stress that this is not licensed for use in the UK. And Pam obviously touched upon tofersen, which was very recently approved by the FDA, which is a very exciting milestone in MND research. So I've just shown some examples of some famous faces who have battled or are still battling MND. So you'll notice on the top left here, so astrophysicist Stephen Hawking. And on the top right, we have Lou Gehrig, so the American baseball player. So over there, they refer to MND as Lou Gehrig's disease. On the bottom right here, we have an American soul singer, Roberta Flack, who was diagnosed a few years ago. And the bottom left picture here, so as Pam also alluded to, says that a very powerful image taken of Rob Burrows, so the former rugby player. This is him being carried across the finishing line of the inaugural Rob Burrow Leeds Marathon, which took place in Leeds last year. So you may remember the ALS Ice Bucket Challenge. So this was raised a significant amount of funding and awareness for motor neuron disease. So what do we know about the biology of MND? So in healthy individuals, we have these motor neurons, okay, which stem from the brain, so from the central nervous system, and they innervate our muscle cells all over our body. And as they attach to the muscle cells, they form what we call these neuromuscular junctions. I'm just going to get a laser pointer here so it's clear what I'm referring to. So when signals are sent down the motor neuron to the muscle fiber, it triggers the muscle cell to contract. Now, in motor neuron disease, a widely accepted hypothesis is that these motor neurons actually die back, so they no longer attach to the muscle fiber. As a result, the signals sent down the neuron rather, do not actually reach the muscle. Therefore, the muscle cannot contract, and as a result, this can lead to muscle wasting, paralysis, and death usually occurs by respiratory failure as a result within, as I say, 2 to 5 years from the onset of the first symptom. So motor neuron disease really is underpinned by genetics, and so basically just to put this into context, so the average human has approximately 20 to 25,000 genes within their genome that make us who we are. And over the course of the last 3 decades or so, we've seen more than 50 different genes being identified as contributing to the development of motor neuron disease. As Pam referred to in the case of the tofersen clinical trial, so this was targeting the SOD1 gene. So this was the mutations in SOD1 were the first to be described, and this was back in 1993. There was quite a long hiatus before the next mutations were identified in a gene called TARD-BP. And in 2011, mutations were identified in a gene called C9orf72, which to date is the most common genetic cause of motor neuron disease. So as you can hopefully infer from this timeline, what we've seen over the last few decades is this almost exponential increase in the number of gene mutations being identified, and this is in part down to the development of more sort of advanced sequencing modalities that we have at our disposal, but also the sort of accelerated international effort that we've seen in obtaining patient biosamples and studying the sequences of their genome. So over the past 7 years, so since I began my PhD, my research has really focused, largely focused on the sort of study of three new MND genes. So the first was mutations in a gene called GLT8D1. And then the following year, we published work on a gene called CAV1. And most recently, we discovered mutations in a gene called CANK1. So this is going to be the focus of my presentation moving forward. But before I go into the details on those 3 genes, how do we actually identify these new, disease-causing mutations? So the best way to really describe this is to, we sequence the genome of patients and healthy individuals, so individuals without MND. Once we sequence all their DNA, we can feed this information into specialized machine learning models, which can detect subtle differences between the MND patients and those living without MND. Now, the problem with this is that we have, what we call a lot of heterogeneity in the population. So, everyone's genetics are different. So how can we prove that a change that we see in motor neuron disease patients is sufficient to actually drive the development of their disease? So we can interrogate this in 2 main ways, really. So we can use bioinformatics. So this is sort of computer-based machine learning algorithms which can predict whether or not a particular mutation is sufficient to cause the disease pathogenesis. Now, I'm not a bioinformatician, so my focus really is on the lab experiment side of things. So this is going to be the focus of my talk moving forward, and hopefully I'll explain to you how we can actually determine whether or not a mutation is sufficient to cause motor neuron disease just using experiments we have at our disposal in the lab. So in my opinion, this is probably the gold standard model that we have available. So what we can do is we can take skin cells from patients living with motor neuron disease. We can then turn these skin cells back in time to what we call stem cells. So these are cells which are present at conception, so at the fetal stage. And this sort of reversal process, if you will, this was discovered by a Nobel laureate called Shinya Yamanaka, I think in 2006. Once we have the stem cells growing in a dish, we can then turn them into any cell type within the body, but obviously in the context of motor neuron disease research, a lot of the groups around the world are focused on the generation of these motor neurons. So from taking the skin cell to actually generating these motor neurons, this process probably takes on average about 3 months, I'd say. And the benefit of using this technology is that we can grow the motor neurons to the brain cells from the patient without the need to take a brain biopsy, which is obviously a very invasive and dangerous procedure. And importantly, these motor neurons retain all the genetic characteristics of the patient from which they came from. So what I've shown underneath here is just what these cells look like down the microscope. So you can see here the stem cells sort of organize themselves into these compact island-like formations. And on the bottom right here, you can see these motor neurons form these beautiful networks, which is reminiscent of what we see in the human brain. So we don't always have patient skin cells available, especially if we identify a new disease-causing mutation. But this isn't necessarily a problem, okay? Because what we can use is this. We can take advantage of this technology called CRISPR. Okay, now CRISPR is a naturally occurring defense mechanism that bacteria use to fight off viral invasion. And so Jennifer Doudna and Emmanuel Charpentier won the Nobel Prize in Chemistry a few years ago for discovering that this naturally occurring defense mechanisms, this CRISPR technology, can be reharnessed to edit the DNA in any species. So think of it like a pair of molecular scissors. So using the CRISPR technology, we can make precise cuts at any point within the genome, and we can provide instructions to introduce a mutation which we want to study. So we can do this in healthy cells. So just going back to the timeline. So as I mentioned, the focus of my talk is going to be on these three genes. And I'm just going to start by sort of discussing my work on CANK1 and how I use that CRISPR technology to sort of study the effects of mutations within this gene. So as I mentioned, we don't always have the patient skin cells available. So we took skin cells from a healthy individual. We then reversed them back in time to stem cells. So as I previously described, and then I used the CRISPR technology to introduce mutations within CANK1, so within the gene, which reflect those that we discovered in the patient population. Once I'd introduced those mutations, I then turned these stem cells into motor neurons, and then I studied what effect the mutation had on their ability to survive. Now, as you can see here, what we did see was the motor neurons became toxic and they died as a result of these mutations. So this tells us that the mutations that we discovered in the patient population are sufficient to cause motor neuron death. Now, moving forward, so we have to sort of put CANK1 into the bigger context. We need to really understand the underlying biology of what is actually going on here. So CANK1 is a gene which is important for transport within the cells. Now, all the cells in our body rely on a very efficient transport mechanism in order to get energy supply to different parts of the cell in order for the cell to function and survive properly. Now, we know that CANK1, this is based on previous literature, is integral to this process. So we can infer from this that mutations within CANK1 are disrupting this transport machinery, and as a result, that causes the cell to die. Now, this already adds to an existing mountain of literature concerning the transportation of cargo within the neurons. So simply providing information on this new gene and how mutations within it can affect this process, it may influence researchers around the world to sort of look at the transportation of this cargo in the context of CANK1. So it might change their approach slightly, which is really useful for the field. So I mentioned the two other MND genes which I researched. This is prior to CANK1. So these are GLT8D1 and CAV1. Now, at the cell, we have what we call the cell membrane. Okay, so this is the boundary around the edge of the cell. Now, within this membrane, specialized regions called lipid rafts form. Now, I'm going to explain a little bit more about what lipid rafts are in the next slide. However, in my own research, what we've discovered is that both CAV1 and GLT8D1 are essential to the formation of these lipid raft domains. And this is important for specialized signaling pathways, which are important to cell survival. Now, over the last few years or so, I've been collaborating with Professor Brian Head. So he's based at the University of California in San Diego. And his team have developed this gene therapy approach, which is called SynCav1. Now, SYNCAV1 is a gene therapy which is able to target these lipid raft domains. So if we take two genes, for example, so here, for example, CAV1 and GLT81, and we identify that mutations within these are sufficient to disrupt these lipid raft domains, we also have a gene therapy at our disposal which can hopefully rectify this process. And this is going to be a big focus of my Motor Neuron Disease Association fellowship moving forward. So what are lipid rafts exactly? So as I mentioned, you have the cell, so the neuron, and you have this boundary around the edge of the cell called the cell membrane. Now, within this membrane, you have lipids, proteins, and cholesterol, which exist in this sort of free-flowing liquid-based environment. Now, every so often, these lipids and cholesterol will be recruited, and they'll cluster within specific regions of the membrane, and they will form what we call lipid rafts. Now, when the lipid rafts form, they recruit specialized signaling molecules to enable a sort of a downstream cascade of signaling events which promote the survival of the cell. So there's a lot of literature now coming out, and this is again going to be the focus of my work moving forward, to suggest that this signaling process in the context of not just motor neuron disease but other neurodegenerative disorders is disrupted. And my belief is that this is due to the fact that the lipid rafts are not forming properly, and as a result, you do not get recruitment of these specialized signaling molecules. So the signals do not reach the interior of the cell, and as a result, the cell dies. So what does this mean for patients with motor neuron disease? Well, the more we discover these new genes and their mutations, the more we can understand regarding, this complex genetic architecture. So, that's the title of my presentation, is sort of piecing together parts of the MND puzzle. There's still a lot more genes, I'm convinced, to identify which might be contributing to the disease. And when we identify these new genes, this can unlock new disease biology, so new pathways, or especially in the context of my own research, we may be able to identify links between different genes that actually converge on the same pathway. So in the context of GLT81 and CAV1, we both see them sort of converge on this lipid raft hypothesis. So obviously, as we identify new genes, we can, and new pathways, this will help us to identify new parts of the cell which we can target with sort of disease-modifying drugs. And this may be, we may need to manufacture new drugs. Obviously, the process of doing this is time-consuming. I think it takes, on average, a decade to get a new drug from conception into clinical trials, or certainly to get licensed, and can cost upwards of hundreds of millions of pounds. So there's a big international effort right now to repurpose existing drugs, which are otherwise used to treat other disorders. And the more we understand about the biology of motor neuron disease, the more hopefully we'll be able to identify other drugs which are already licensed and which may be able to provide therapeutic benefit. So what is next for me in my research? So as I mentioned, I recently secured this MND Association fellowship, and the key question I really want to answer here is whether or not lipid rafts are sufficient to drive motor neuron disease. So, in order to answer this question, I'll continue collaborating, with the Brian Head's team at the University of California in San Diego, and it is my theory that if we can sort of discover that these lipid rafts are broadly affected across the entire genetic spectrum of motor neuron disease, so they're disrupted regardless of the mutation the patient carries, then these will be a very interesting and exciting therapeutic target moving forward. In terms of my personal development, as I say, I'm on this independent research track now, so I'm going to be in the process of applying for more research grants with the aim of establishing my own lab group, sort of specializing in lipid raft biology, and hopefully this will happen over the next few years. So obviously, this work has not been possible without the immense support of collaborators, both within the University of Sheffield and of course further afield. Various streams of funding have been integral to the success of this work, and of course none of this is possible without the generosity of patients who have kindly donated their biosamples to enable this research to happen. So thank you all very much for listening, and I'll happily take any questions.
Thank you. Pam, over to you.
Okay. Okay, thank you very much, Toby. That was nice and clear. So shall we look at what is in the chat first of all? So the first one, which I think is also in the pre-submitted questions, is from Mary Luke, who says that my late husband had MND and our daughter has multiple sclerosis, and has any connection been found between these two conditions? Toby, are you happy to have a first that?
I'll have a go. Okay, so obviously I'm not an MS researcher, but from what I understand, there's no convincing evidence of an overlap between MS and motor neuron disease. So MS is obviously an autoimmune disorder. So this is where the body's own immune system will selectively attack and destroy what we call the myelin sheath. So these are the insulating cells around the neurons. So as a result, what this does is it slows down the sort of conduction of these signals, as I mentioned, along the neuron, which sort of delays, the sort of increases the latency period in which you're able to move and respond to things. Now, in motor neuron disease, it's the neurons themselves that degenerate, and more specifically the motor neurons. So yeah, I hope is that accurate enough, Pam?
Yeah, I think there isn't a. We do have a small collection of patients who have had very long-standing multiple sclerosis and then have gone on to develop MND on top of that after many years. But that is rare, and it's not a usual thing that you would expect to happen. So it's rare for a person to develop both disorders, but there's a very small collection of cases where that can happen. So let's go on. I hope that answers your question, Mary. So Angelica was asking, will a recording be sent to the audience? And I think Helen mentioned that, yes, it will be shared with you all. Then we've got Effie Callie, who asks, do we know the causes for MND generation, e.g. air, food, drugs, etc.?
Okay, so again, I'll give this a go. So what we do know, obviously, I've hopefully convinced you in my presentation that it's really underpinned by genetics. Okay, so certain mutations are sufficient to cause MND in individuals. But what we do know is that there's a high degree of sort of environmental influence. So obviously, as Pam alluded to in her presentation, exercise we identified is a risk factor for the development of MND, but if you have an underlying genetic mutation, so a predisposition to it. And there've been various things, other things linked to the development of MND, but it's my thought that there's usually a genetic underpinning and a risk associated with this, and I wouldn't like to say it's purely environmental or genetic. There's probably an overlap between the two, so it's quite complex, but very good question. I don't know if you want to develop on that, Pam.
Yes, I think so. Between 5 and 10% of MND patients will report someone else in the family that's had the condition. So that's clearly a strong genetic influence. But 90 to 95% of patients do not have a family history. But even in those so-called sporadic MND patients, we think that genetic factors are important, but you may have to have 4 or 5 susceptibility factors that when they come together, they make your motor neurons more delicate, less able to repair themselves over the years. But if you have to have 4 or 5 factors, you're not going to pass all 5 of those susceptibility factors to any one of your children. So it doesn't appear genetic in the traditional sense. But we've studied the commonest genetic subtype of MND that Toby mentioned, caused by changes in the C9orf72 gene. And what we found is people can carry that gene and live perfectly healthy lives until they're 50 or 60. And what we found, we've got a way of measuring strenuous athletic activity, and the stronger your athletic activity over your adult life, if you're a carrier of that gene, the earlier the MND declares itself. So there's a sort of complex genetic environmental interaction. And, you know, you can carry a risk gene, but then there are a number of steps that need to happen for the disease actually to declare itself. And one of the things we're very interested in is, so how can a person carry one of these risk genes and be perfectly healthy for 50 or more years? So we're trying to, and clearly there have been compensatory factors that the person is able to mount to keep themselves healthy. And so we're trying to understand, so what changes for the disease to manifest itself? And if we can understand those compensatory mechanisms, that might give us a fruitful list of therapeutic approaches. So it's a really good question, but difficult to unravel. So we're trying to mimic strenuous physical exercise in the cells that Toby has talked about in the laboratory. So exposing them to the stresses that a rugby player or a marathon runner might expose themselves to when they're doing that really strenuous exercise. We're careful though, we're cautious, because clearly exercise is usually a good thing. It's very good for your cardiovascular system, preventing certain diseases, cancers even, and Alzheimer's and Parkinson's. You know, doing exercise might be a good thing. So we don't want, until we understand this fully, we don't want to dissuade people from doing healthy exercise. Very good. So let's see what's next. So Dariush has said, is this disease a problem of the modern era, or has it existed in past centuries? And another question is whether the disease is increasing. Toby.
Okay, so yeah, so a good question. So obviously it's something that, you know, was only just, I can't remember the first date it was described, to be honest.
1869.
Fantastic. There we go. Yeah. So this, it's not to say that it didn't exist before then, of course. So obviously, you know, it's the way sort of diagnostic tools have developed. I'd say, you know, perhaps before that, you know, cases are often misdiagnosed as something else. And in terms of, you know, are we seeing more of it now? What we do have is an aging population. So people are living longer now, and motor neuron disease is an age-related disorder. So we will, as a result, see an increase in the number of patients being diagnosed. And this is also true of other neurodegenerative disorders, you know, see Alzheimer's and things. So yes, it's I'd say it's always existed, but the prevalence rate is increasing.
Very good. And then there's some nice comments from Jennifer Pinder and Dennis. They like the talk. And then Muriel Akam has asked about possibilities of gene therapies.
Yeah, so yeah, gene therapy is obviously is one of the main sort of ways people are trying to tackle motor neuron disease. Now, obviously, the more we understand about which genes are involved and the mutations of those, you know, in causing the disease, the more we can look at ways to actually silence those particular genes. It's, you know, it's still ongoing. You know, so, you know, gene silencing is not an easy feat at all, but it's definitely, you know, obviously Pam mentioned the you know, the GTIMC, you know, so manufacturing, you know, certain viruses which we can use to target, you know, these mutated genes is definitely something that's on the, I'd say, on the horizon. Yeah.
Yeah, and just to add a little bit to that, following the success of the tofersen trials that I mentioned to you briefly, there are other genetic therapies in clinical trials at the moment, including for some of the other genes, but also so there's a rare gene that tends to affect young people called fuzz. So there's an active clinical trial for that. We did try two genetic therapy trials for the common C9 gene that causes MND, and they did not work. So we've gone back to the drawing board for those ones. But excitingly, there are some genetic therapy approaches for sporadic MND, so where we haven't identified a single gene, we can target with gene therapy some of the proteins that are contributing to the motor neuron injury. So as motor neurons get injured, they accumulate these protein aggregates of proteins, and the main protein is one called TDP43. So, for example, there are genetic therapies aiming to lower the clumps of abnormal proteins that are forming. So I think it is a very exciting era that not every gene that causes MND can be tackled in this way, but a good array of them can be. So I think it's going to be a very important way forward in the next few years. And so Ian Molyneux, or Molyneux, I hope I'm pronouncing that correctly, has asked another very good question. Can the same research methods for MND be applied to Alzheimer's and Parkinson's disease?
Absolutely. So yeah, we're doing precisely using very similar sort of approaches, you know, for both of those disorders in Citran itself. So yeah, this also involves, you know, the sort of taking patient skin cells, so what I described, and growing the neurons as a result. This is certainly something we do a lot of in the context of Parkinson's disease. So yes, the same research methods can be applied, and you know we do we do it routinely. So yeah, not much else really to it.
And I guess, you know, the impetus for creating Citran really came from MND patients and their families, but quite deliberately, we didn't want to make it a research institute that only worked on MND. So we have teams that work on Alzheimer's disease and the aging brain, Parkinson's disease, stroke, and so on. And we learned from each other because there are the mechanisms of neuronal injury. There are common factors in all of the major neurodegenerative diseases. For example, the mitochondria, the batteries in the cells, go wrong or aren't functioning properly in Alzheimer's, Parkinson's, and MND. So we have kind of shared programs looking for drugs that can correct the function of those cellular batteries, the mitochondria. So very much so. That's a good question, Ian, and we are very much applying that. So then William van Roosven says, thanks. How long might it take for repurposed rather than new drugs to get approval for this disease?
That is a question I don't think I have the answer to be honest. Obviously, so we mentioned the drug screening facility that we have in Citran. So this is obviously used to screen sort of libraries of these drugs available, which are already licensed to test whether or not they are of therapeutic benefit within our models that we develop in the building. Now, my guess is it'd be very rapid if we get these positive hits, but in terms of time frame, I might have to refer that to you, Pam. I'm not entirely sure. Yeah.
So yes, we have taken every FDA approved drug for human use, and we've done an analysis to be able to predict which of those drugs would get across the blood-brain barrier into the nervous system. So it's no good for a disease like MND working with a drug that doesn't get into the nervous system. So there are about 550 of those drugs known to be safe in humans, and we're screening that library in our models of disease, particularly the human cellular models that are generated from skin cells, as Toby described. So just to give one example, we did that in Parkinson's disease, where the batteries, the mitochondria in the cells that generate energy aren't working properly, and we screened that library to look for drugs that are known to be safe in humans that improve the function of the batteries. And there's one called ursodeoxycholic acid, UDCA, that had been used in humans for liver disease, but improved the energy generation in skin cells from Parkinson's disease. And we were able to take that straight across the road into the Royal Hallamshire Hospital into a phase 2 trial in Parkinson's disease patients. So the first phase was a study with 40 patients, but we were able to show that the walking, the mobility of patients that were on the active drug compared to the placebo drug, that we have to test new drugs in that way. The walking function was better in the patients receiving UDCA, and we have a sophisticated imaging method whereby we can measure the energy currency, the ATP, in the living brain in human beings, and we could show that the energy levels in the brain improved with UDCA. So that's now going into a phase 3 trial in a bigger number of patients, and we hope that that will come through as a treatment. So that's one example, and we've got things coming along. We also showed that UDCA seems to have a positive effect in cells from Alzheimer's disease patients as well. And we're just testing it now in the cells from MND patients. So very much so, it's a much more nimble way of getting therapies into the clinic if they've had all that expensive safety and toxicology testing beforehand. So we're very interested in repurposing existing drugs. So then James Milligan asks, do we know why some patients follow an atypical clinical course, initially rapid, but then progression slows and halts? Is there a genetic theory or basis for such phenotypes? Again, a very good question, James.
A very difficult one for me to answer. Obviously, I'm not a clinician, but there's this actually links to a question further down. So is it from Avril Aslett Bentley, who asked, is it likely that some sufferers from MND have more than one of these genetic mutations that make the condition worse? So so this would actually perhaps accelerate the disease process. Now, this is actually something I've been researching in the last year or so. So I mentioned obviously the genes C9orf72, so the most common genetic cause of motor neuron disease. And what we've identified is additional mutations in some patients, and they appear to have a more rapid disease progress as a result. So in my own work, we then use CRISPR, the technology I described before, to then model those additional mutations. So introduce those additional mutations into cells from the C9orf72 patient. And we looked at the effect it had on their survival, and indeed in the lab setting, just like we saw in the patient population, it makes it worse. So yes, there is genetic evidence now, available to show that is the case. I don't know if you want to develop on that.
Yeah, no, that's a good answer, Toby. And we are, so we're taking biosamples from all our newly diagnosed patients, some of whom have slower disease progression, some more rapid disease progression, to try and understand. So if we take the example of Stephen Hawking, the professor of astrophysics from Cambridge University, he developed MND in his 20s, and he had it for more than half a century, more than 50 years. And clearly he was very disabled at the end of his life. And I guess his life was extended by respiratory muscle support. So he had breathing devices to help his breathing muscles. But clearly he had very slowly progressive disease over decades. Some people run into big problems within a few weeks of noticing the first symptom. So if we could, so one of the other things we're trying to understand from donated patient biosamples, so we can take skin cells from patients with rapid or slow disease progression, turn them into motor neurons in citran, or turn them into the neighborhood cells that sit around the motor neurons, and then interrogate all the genes expressed in those cells. We can use RNA sequencing or transcriptomics, as it's sometimes called, to look at which genes are up and down regulated in their expression to help patients keep the disease at bay with slow progression. And if we could turn the rapid progressors into slow progressors, that would also be a great step forward. So we're using all these techniques of modern neuroscience to drill down into the detailed gene expression and biochemistry in the cells, trying to understand differences and then design new therapeutic agents, either small molecule drugs or genetic therapies, to help with treatment approaches. And I think understanding slow progression would be really helpful in that regard. So next question.
Probably the last question, Pam, I think.
Last question, yes. So it's from Isona Collins. Are there opportunities for MSc graduates at SHA to join this program? Yes, very much so. We have lots of opportunities for either graduate technicians, so a lot of people who've done a master's course, not sure whether they want to spend 3 or 4 years doing a PhD, and a good way of getting the feel of that is perhaps to do a graduate technician post for a year or 2 and then make a decision about PhDs. If you're certain you want to do a PhD, then there are PhDs advertised all the time on the Citran website and through the flagship Neuroscience Institute and so on. So very much, yes, we're very welcoming of master's students coming to join our research teams.
Thank you very much indeed.
I think there's a lot more questions. 45 new messages, I think. I'm sorry we've not got time to answer them all, but I think maybe Helen, do you want to, or Andrew, do you want to say something about those people whose questions we haven't been able to get to?
So we'll share all of your questions. We'll take a copy of the chat. We'll share all your questions with Toby and Pam, and you will receive much more information in terms of what we are doing about some of these neurodegenerative diseases. And it's going to be a big focus for our communications plan over the next 12 months. So hopefully you'll get answers to many of the questions that we haven't got round to today. Just a huge thank you to Dame Pam, to Toby for sharing with us your inspiring research, and to Pam for your engaging introduction, and also for facilitating the questions and answers. Thank you to all of you online who may have already got involved and supported the research at the Sheffield Institute for Translational Neuroscience. You really do help our work and help to support the lives of patients living with MND and other neurodegenerative diseases. Thank you to the alumni team for running this event for us today. And if you were inspired by today's event and the amazing research that people like Pam and Toby are involved in, you may be wondering how you can support them and others to continue this work. And really, there are several ways that you can do this. So you can make a donation to the MND research by visiting sheffield.ac.uk/giving/MND. And as Pam said, if you aren't able to do that yourself and you have contacts and connections that you may want to share with us, please do so. And you can email the alumni team. If you're in Yorkshire, the Midlands, the Northwest, you can pop into a Thornbridge pub over the next couple of months, where they are going to be offering a big walk beer. So those, 10 pence of every pint drunk will be sent to MND research at Sheffield. Or you can also participate in the big walk, which is happening this year on the 28th of June. There are many other ways as well that you can help, and please do contact the alumni team if you are interested. So a final thank you goes to all of you for participating in this event. There will be a follow-up email. We'll give a link to this video. If you'd like to look at other webinars in the series as well, please do visit the Bright Minds webpage. All of our research should be showcased on the Sheffield Player, which you can visit by going to player.sheffield.ac.uk. And we'll share details of that also in the post-event email also. And please do, if you can, take a minute or two to complete the short feedback survey. If you do want any more information on what you've heard today, please do contact the alumni team. Thank you once again for attending the Bright Mind session. And whether it's morning, afternoon, or evening, wherever you are, I wish you a great rest of the day. Thank you very much indeed for attending. Thank you.